Hi, my name is Georgia Ray Atkins and I'm a Sizer Scholar. I'm a senior cell and molecular biology major and I work in Dr. Gail Cornwall's lab. My current project is characterizing the epididymal matrix. Let's take a look. Okay, let's take a tour of our main lab. So this is our biggest area. We always have music going, it's really lively, get a lot of work done. But we all have our own lab bench with our stock solutions. We have various lab equipment, a couple freezers, a fridge. This is our vent where we make, toxic chemical solutions. We also have a cell culture hood back here, which I'll show more about cell culture a little bit later. We have some incubators, water baths. This is, just an extra bench area. And then over here is where I work and our grad student work. So we each have our own lab space and then also our own desk. We have a couple tabletop centrifuges for our use. And then back here is where we do stock solution prep. So we have all our chemicals, dishes. And then this last lab bench is a common lab bench that we use to run various gels and then also prep for our Western blots. All right, and here we have Keegan working on our cell culture. She works on them in the hood and then she moves over here to count them on our microscope. And if you can see right next to her to the right, then that's just where they grow. Okay, now I'm in our secondary lab space, which isn't as lively as our main lab, but we don't use it all the time, so that's okay. We have many important pieces of equipment in here that we use. The first one here is actually our protein prep machine, which allows us to make our own proteins. We don't have it in the main lab because we want to keep it protected, and also we don't use it all the time. But you have to go through extensive training to learn that, but it's really useful once you're able to. And then over on this side, it's just more common workspace that we have if we need overflow from our main lab. And then if we walk back here, this is where I do a lot of my project work. We have our own microscope, and I'm able to look at my sample with this scope and then also take pictures, which is why I'm working on the computer right now. And then also in this back corner, we have something called an ultracentrifuge. We call this our baby ultracentrifuge because it's a lot tinier than the main ones. But we're able to extract different samples from this ultracentrifuge. All right, we're gonna walk into the dark for a second. This is our secret tunnel. But this leads to something called our dark room. And if you've ever had experience with Western blots, this is where you expose your Western blot. So you have a little workbench here. And then in this corner over here, you have where the machine that processes the film that you use for your Western blot. And the reason it's called a dark room and why all the walls are painted black is you cannot have your sample exposed to light for your Western blot. So they have these special red filters. And when you turn it off, then it goes completely dark and you're now in a light safe room to do your Western blot. Okay, the last place I'm going to show you is just our confocal microscope that belongs to the department. A confocal microscope is a lot higher power and actually uses lasers to capture the image. So the lasers are these big boxes over here on the left. And then if you look over to the right, here's just what the confocal looks like. It's really high tech. You move around using that joystick that you see on the right. But you get really good images, as you can see here. Here's an example of the software and then a picture I was able to take on the confocal microscope. So we have a lot of different options for also collecting data here as well.